Role of enzymes in pathogenesis. We will see different types of enzymes produced by the plant pathogens and their role and functioning in the development of diseases. My name is NH Shankar Reddy. I am doing second year PhD plant pathology in Annamalai University. So we will start with the definition of enzymes. So enzyme is a large protein molecule which act as a catalyst to bring the specific chemical reaction into the living cells. Actually, this large protein molecule enzymes are ranges from 2 to 100 micrometer in diameter. So this converts the specific reaction into the specific products. So each and every living system will require enzyme to convert the specific reactants into the specific products. Then only the human system, not only the human system, the living system will get the energy. So the conversion of large molecule into the small molecule will be very essential, so that it can be very easy to the cells to absorb the energy. So here all cells contains, each and every cell contains millions and millions of enzymes that are belong to the different kinds of enzyme groups. Let me tell you an example. An average in a human system contains 25,000 to 30,000 genomes. So each and every gene is responsible for the different types of characters. Here in the meantime, here each and enzyme, every enzyme has a different roles and functions in plant system, not only in plant system but also the biological living system. So coming to here, so enzymes secreted by the microorganisms which have a definite functions, which has a key roles in disease development. It has a key roles in disease development. So what is the main function of the enzymes that is secreted by the plant pathogens? Here they break down the large plant molecules, I mean large molecules of substances into the small molecule. So what will happen due to this? Due to the conversion of large molecular complex molecules into the simple molecules, the pathogen can easily absorb and it can get the nutrients whatever the pathogen requires. So the energy requirements will mainly mediated by this enzyme. So due to the conversion of large molecule into the small molecule, the pathogen can easily absorb the energy. So what are all the different types of enzymes which are produced by the plant pathogens? We will see one by one. The first one is cutinases. So cutin is the main component in a plant cell wall. Again, we can see the definition here. Diagrammatic representation above the cell wall as a plant cuticle will be there. So which is the very chief and important component in plant cell wall. So what will happen? This cutinases, the plant pathogens which produce an enzyme cutinases, which degrade the cutin, that is the main and chief component in plant cell wall. So due to this, what will happen? Due to the degradation of a plant cuticle, we can see here in the diagram, we can see in the plant cuticle, it can easily destruct the plant cell wall and enters into the plant system. So this type of cutinases or enzymes are produced by many plant pathogenic fungi and bacteria. I have given one example, that is Puccinia hordi. This is the structure of cutinases which is produced by the plant pathogens. So this diagram represents each and to understand what are all the structures and layer will be there in plant cell wall. So it is a primary cell wall, then middle lamellae, then pectins, lignins, different types of components where the plant cell wall is made up of. So here we can see chitinase, cuticle, chitinases or cuticle is which is the chief component of plant cell wall. So due to the degradation of a plant cuticle, the plant pathogen can easily are able to enter into the plant cells. Here we can see the mRNA or cutinase molecule which is degrades that. This is the diagrammatic representation. The second enzyme is pectinases. So this pectinases is a substance where it is also the chief component of middle lamella. We can see here middle lamella on the right side. This is the middle after the primary cell wall. We can see the middle lamellae. So what will happen due to the production of the pectinases by the different types of plant pathogens? It degrade the middle lamellae of the plant cells. What will happen due to the degradation? This all enzymes have a different types of function. The main aim is to degrade the different types of substances which act as a barrier. So after destruction of this type of substance, it enter into the plant cells and it disrupt the physiological mechanism of plant cells. So pectinase type of enzymes are produced by Phytophthora and Pythium. So these two, not only these two, many plant pathogens also be there, but that I am mentioning the important ones. So Phytophthora and Pythium will produce pectinase type of enzymes. Here you can see the pectin. Pectin will be here. This degrade pectins. This is the chemical composition and all of pectin. The third one is cellulases. So plant cell wall is made up of cellulose. Yes, so it is also one of the major and important component of plant cell wall. So these cellulases are produced by different types of fungi, bacteria, nematodes, which has a main role of softening. Softening means it making of simply degrading. So due to this, what will happen? Due to the softening, the plant cell wall lose its stability and capability to protect from the harmful pathogens. So due to this, what will happen? The plant pathogens can easily able to enter into the plant system. So this type of enzyme is produced by fusarium. So this is the structure of cellulases and the chemical structure composition. So here what will happen? Cellulose, the one of the chief and this all primary cell wall or secondary cell wall, this all cell wall is made up of cellulose. So due to the destruction, what will happen? The pathogen enter into the plant system. Next one, hemicellulases. So these hemicellulases are the complex mixture of polysaccharides, which is also the chief and important component of plant cell wall. So this hemicellulases which break down the hemicellulose, which is also the chief component of plant cell wall. So this type of enzyme is produced by sclerotiana, where Anton de Barry initially worked on sclerotiana and discovered cell wall degrading enzymes as well as on other systems. So here we can see the diagrammatic representation of sclerotiana. This is soft rod fungi. The next one is lignases. So break down the most resistant component to enzymatic degradation, that is lignin. So lignin is the most powerful and most resistant to the enzymatic degradation that can be degraded by the enzyme lignases. So this lignases will degrade the lignin. We can see here lignin is here. So this is the primary cell wall, middle lamella, secondary cell wall. Below the primary cell wall we can see the lignin here. So this ligninases will degrade this lignin components. Due to this degradation, what will happen? The pathogen enter into the plant system. So we can see the different types of example. Brown rot fungus, white rot fungus will produce lignases enzymes so that it enter into the plant system. So it simply so degrades. It degrades. Due to this, what will happen? Degrades. It almost lost the strength and stability to protect from the different types of plant pathogens. So so a few of the enzymes are things we given here: pectinases, pectic esterases, hydrolysis, lyases, lignases. What are all the enzymes, different types of groups that have been mentioned here? The next one is proteases. So name itself indicates proteases, which degrade proteins. Proteins are the very important and building blocks of each and every living system. So it degrade proteins. What will happen due to the degradation? The organization and function of the host cell may be lost. The physiological functionings may be lost. So the protein degradation is due to the production of proteases or proteolytic enzymes. So these enzymes will degrade proteases. So example, Helminthosporium virus, that is brownly spot of rice, which produce proteases types of enzyme to enter into the plant system. This is the enzyme structure. Then next one is amylases. So this amylases which break down starch into glucose and utilized by the plant pathogens. So this fatty acid is directly utilized by the pathogen. So it is a fatty acid molecule that can be directly utilized by the pathogens. So why this plant pathogen is producing different types of enzymes to get the energy? So for causing for causing infection or pathogenesis process, the pathogen has to establish itself. So it required the energy molecule. What will happen due to the production of enzymes? It degrade different types of components or substances present in the plant cell or cell wall. What are all the barriers that can protect from pathogens? It will degrade into the complex molecule, into the simple molecule, so that it can get the energy. So after establishing, what will happen? Enter in the plant system and produce toxins. So here toxins plays a major role in disease causing or pathogenesis. So enzymes mainly act as a few of the enzymes also responsible for the playing a major role in disease. So but the mostly toxins plays a major role in disease causing or disease disease causing. So here amylases types of enzymes is produced by many plant pathogenic fungi, bacteria and nematodes. The last one is lipases. So here the lipases will break down the lipids like phospholipids or glycolipids. These are the energy sources or energy materials for plants also. So the energy material can be stored in the form of lipids. So it can break down the lipids into the small small complex lipids into the small molecules, so that it can be utilized as the energy sources for pathogens. So it is produced by several types of fungi, bacteria and nematodes. So here we will see example: Sclerotium, Ralphium, Botrytis cinerea will produce uh, lipases enzymes. So these are all the different types of enzymes in pathogenesis.